What is a good target for this genetic engineering? We have different cultivars out there, and from the turf perspective, Argentine behavior grass is a preferred turf. It has some other benefits for genetic engineering and for genetic improvement. The benefits are that it produces a uniform seed progeny. So every seedling is identical, and it does not transfer transgenes by pollen very efficiently. So we have studied this in another experiment up there in Mariana, where we confirmed that the gene transfer rate by pollen, even in a low distance, is less than 0.03%. So that means we have a situation where we have a high safety compared to sexually outcrossing plants. Now we introduced the two different transgenes. One of them is a growth regulator, the pathway for a growth regulator. So that growth regulator is now modified in the transgenic plants. That means the plant itself, as well as all the seeds produced from the plant, have this modified pathway. We do not have to introduce any chemical to maintain this genotype. It's genetically modified, so that means the plant itself does what we usually do if we apply growth regulators. And this particular growth regulator, what it does is enhances the outgrowth of axillary buds which form new shoots. That increases shoot density and to some degree will also reduce the plant stature. If you look over here, you see the wild type Argentine behavior grass. Then this is a transgenic line which shows some degree of dwarfing and a few more tillers. And this is the extreme behavior grass transgenic line, where you see which doesn't even look anymore like a behavior grass. Obviously, this is just here to exemplify how extreme these differences can be and how big the difference are from transgenic line to transgenic line. And these differences are caused since making these transgenic plants. Some have few copies of the transgenes, others have many copies, which changes the amount of the protein that changes the plant pathway for this growth regulator. Now, obviously, a question comes whenever we change the plant architecture, and that's why we wouldn't like this ultra dwarf. We somehow may also affect its drought tolerance, which is one of the prime parameters that we enjoy with behavior grass. And therefore, we have established a study here where we look into irrigation requirements of this modified behavior grass. The transgenic lines which we favor are marked in this study with a red flag. The wild type behavior grass, which is the one that you can buy in the store, is marked with a white flag. And then we also have St. Augustine grass in this study to just see how it performs under these low input conditions. So you can look at these different growth conditions that we have in this environment. We have three different environments. The environment that you see right behind of me is an environment which has very low irrigation frequency. Then there is a non-irrigated treatment in the far back. And then there is a heavily irrigated treatment in the middle sections. We have every environment repeated 2 times. And we have 2 different mowing regimes, since mowing also affects to some degree drought tolerance. And the homeowner typically would prefer not to mow the lawn too frequently. So here on the right side, we mow always every week. On the left side, we mow every 2 weeks. And the mowing height is 3 inches. The amount of irrigation that goes into the moderately irrigated plot behind me is approximately 1/3 of an inch once a week. So it was just irrigated today. So you can see how it looks right after irrigation. But you can already notice that the St. Augustine grass looks stressed, whereas the transgenic as well as the wild type behavior grass performs very well and doesn't show any sign of stress. This is how we adjust the irrigation treatment. Now, the irrigation is controlled by soil moisture sensors that are in the ground under the plots, and these are connected to a computer-controlled system which triggers irrigation. So the irrigation setup that we use are the mini wobblers, which irrigate very consistently and uniformly, and they allow us to maintain a situation where we can evaluate what level of irrigation is necessary to maintain the behavior grass in a good growth condition, and we can see how it differentiates from the St. Augustine grass. Whereas in the middle plots, we have an irrigation treatment that is necessary to maintain good quality in St. Augustine grass, and then we can see what the difference in irrigation requirement is. So if you compare now the different transgenic behaviors in trees here with the wild-type behaviors, what you will notice is, and the transgenic line that we prefer is flagged with a red flag, the wild-type is flagged with a white flag, and you will see that the Argentine behaviors that is not transgenic has a more open growth habit, is kind of a little bit more sparse looking, similar to the behaviors surrounding us, but not as severe, since Argentine is a better turf type. Nevertheless, the plant grows more horizontally, not as much upright, since it doesn't have as many tillers. So the biggest difference in these grasses is the number of tillers that the plant produces, and that is a result of this growth regulator. If you look into the non-irrigated study in the very far back on your way out, you will notice that Behair grass doesn't perform very well under the current drought if it's not irrigated at all. However, the transgenic outperforms still the wild-type Behair grass, so it becomes obvious that the transgenic modification does not compromise the drought tolerance of our Behair grass, and St. Augustine grass in this study is almost degraded, so there is hardly any green leaves on these plants. You can see it from the far distance. The ones that look very yellowish, this is the St. Augustine grass out there. So after completing this experiment, we concluded that this particular trait is very favorable. Now, for commercialization of this improved grass, it would require deregulation by USDA aphids, which is a time-consuming and costly process. And because of that, we have engaged in targeted mutagenesis and chemical mutagenesis, which, although they are capable of producing very similar results, are not subject to deregulation. So this study will go into the ground over there on the other side, and we are hoping to find a similar response. Also, what we noticed in this study is the plants still produce seeds and seed head, but the seed head production is delayed. So in general, we use this genetic transformation technology to identify and confirm targets for genetic improvement, and then subsequently target the same genes by mutagenesis, which is not subject to deregulation.